Going to deal with dermatophytes. Dermatophytes are group of group of keratinophilic keratinophilic fungi, which means keratin lover, keratin lover, keratin loving fungi, lover or loving, and like it's in fact keratinized tissue like tissues like skin, hair, nail, and nail. It is like causing superficial mycosis. Cause superficial mycosis. Mycosis known as dermatophytes. Mycosis known as dermatophytes. These are superficial mycosis, or it's also known as tinea infection or ringworm. Based on the classification, like there is a classification of this classification based on morphology or morphological classification. When we look, there is one trichophyton, it's in fact like hair, skin, and nails. There another one is microsporum. It's in fact like skin and hair. And then third one is epidermophyton. It's in fact like a skin and nail. And there is another classification based on the habitat. Like it is called ecological classification. In ecological classification or habitat, based on the habitat, like it is three type: anthropophilic, like in fact only human. Another one is zoophilic. It's in fact primary animals, occasionally humans. And then another one, geophilic. It means from soil, found in soil. When we look some example of this habitat, so anthrophilic are like trichophyton, rubrum, then epidermophyton, plasticum, Epidermophyton flocosum. When we look to zoophilic, there is a microsporum. Microsporum canis. And then when we look for the geophilic, there is a microsporium. Microsporum. Gypsum. Regarding how it's transmit or infect person, regarding pathogenesis of this disease, this infection, like there is transmission from direct contact like who is infected person or animal or soil that invades, like invasion is limited in due to its only infects keratinoid tissues do not penetrate in the deeper due to the presence of immune response do not deeper do not go due to the immune response, but in keratin layer not immune response presented. So now in that, after the infection, there is like dermatophytes. This mechanism, dermatophytes secrete keratinase enzyme which digest keratin. And there is immune response involvement. There is also immune response involvement. Cell-mediated immune response is present in this case, and that lead cause to inflammation. So now we are going to classify as clinical manifestation. So clinical manifestation in this disease, clinical manifestation of dermatophytes tinea infections. There is some tinea like tinea capitis, which infect site scalp, and the organism is Microsporum audouni. And there is another tinea corporis, just no site and which the tinea like body, side is body, tinea ringworm. And after the tinea cruris, groin region. When we look for tinea pedis in legs, tinea pedis, tennis pedis in leg region, foot region, athlete foot from foot. Now tinea ungum, tinea ungum. It means it's affect nails. Onkai and como is mycoses, unicomycosis. Then tinea barberi, In tinea barberi, it's a beard region. When we tinea fasciae, tinea fasciae, tinea fasciae, like face region, excluding beard area, So when we look in like this, in the scalp region or head area, there is like tinea corporis. So there is tinea capitis. Corporis is like body region in the body part, under muscle. When tinea cruris is the groin region, inside the groin region. When tinea pedis in the foot region. And when look in this image, there is in the beard, there is tinea barberi. Then there is tinea crurisi in the groin region. And when we look for this is hand, palmar tinea paginum. Then tinea corporis in the leg region. Tinea corporis. Then we look for tinea pedis in foot region and tinea capitis in the scalp region. This is regarding clinical manifestation tinea. So now we are moving on to laboratory diagnosis. Lab diagnosis. In the laboratory diagnosis, there is a specimen collection, like a skin wrapping, a specimen collection, like a skin scraping, hair blocking, or nail clamping, clipping for lesion margin. There is direct microscopy. Regarding this direct microscopy, there is a QS mount. 20%. There is in direct microscopy, there is separated hypacine. And orthoconidia arsine. When we go for the culture, there is sap broad testocagar, and it's grow in 1 to 3 weeks. When we look for morphology, there is pigmentation. Pigmentation help in identification. There is lactophenol cotton bloom. Another is culture is lactophenol. Lactophenol cotton blue P CB mount. Here we can see like microconidia and macroconidia. Help to represent genera. There is wound. There is one another test. Evidence woods lamp UV light on light. There is some in this test. There is some fungus or mycoses dermatophytes, which you can say like canis. This is regarding laboratory analysis. So this is regarding dermatophyte